Okay, let's take a look. It looks very complicated, but in this slide, we just focus on the number of carbon atom. Let's take a look. We have 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon. Okay, the 5 carbon. What is the name of this 5 carbon molecule? RuBP. Okay, this is a 5 carbon molecule. We call it RuBP, but we have 3 of them at the beginning of the Calvin cycle. So that's why it says in here 3 times 5 equal to 15 carbon. And then we add 3 carbon dioxide, and then we use the RuBisco. Remember, this step is the carbon dioxide fixation. This is carbon dioxide fixation here. Uses the enzyme RuBisco. You may call it RuBP carboxylase. Then adding the 1 carbon into the 5 carbon, you'll get what? 6 carbon molecule. And the 6 carbon molecule will split into 2 3 carbon molecule. Remember the name of the 2 3 carbon molecule? It is called 3-phosphoglycerate. So you have 3 carbon atom here. And you got 6 of them. Because after you add the carbon dioxide into RuBisco, after you add the carbon dioxide into RuBP, not RuBisco, sorry. After you add the carbon dioxide into RuBP, then they become a 6 carbon molecule, and then we split the 6 carbon molecule into 2 3 carbon molecule. The 3 carbon molecule is the 3-phosphoglycerate. So that means 3 times 2 equal to 6. The 3 here is the same 3 here. Okay. 2 because we split the 6 carbon molecule into 2 3 carbon molecule. Okay. We split the 6 carbon molecule into 2 3-phosphoglycerate. That's why we have 6, total 6 3-phosphoglycerate. And then after that, the 3-phosphoglycerate go through the reduction, carbon dioxide reduction that require ATP and NADPH from the light reaction. Since we have 6 3PG, we also need 6 ATP and 6 NADPH. Then what do we get at the end? You get G3P. And we also get 6 G3P, okay? Because G3P also has 3 carbon. 1, 2, 3. The 3 carbon is the same 3 carbon from the 3PG. So the number of carbon atom is the same. 1, 2, 3. It's the same carbon atom. Well, we got 6 G3P. Then what happened? One of them is used for making sugar. One of them is to make a sugar. That's why it says that, to make sucrose for transport, starch for storage. Basically, you can assume that we use 1 G3P for glucose, to make glucose. And then the other 5, what do we use them? We use them to combine the other 5 G3P. We use them to combine with, basically we use them to make RuBP. That's the whole goal. The rest of the G3P, we use them to make RuBP. So this is what we talk about. What 5 plus 3 is equal to 3 plus 5. 5 times 3. 5 times 3 is equal to 3 times 5. This is what we meant. So we have 5. We use 5 G3P. So G3P. G3P again is the 3 carbon molecule. That means 5 of the G3P. Each G3P is a 3 carbon molecule. That's how we got 15 carbon. Okay. And then we use 3 ATP and ADP so that we have 3 phosphate group. We add 3 phosphate group to them. And then we combine. We use the energy to combine some of the G3P, so that it will generate 3 RuBP. Each RuBP has 5 carbon molecules. So that's about it. Yeah. And of course the ATP here, they come from the light reaction as well. So this is the last step, regeneration of RuBP. So, okay, you just need to know this chemical reaction. That's all I ask you to so-called memorize or know. You have 5 G3P molecule. Remember how many carbon we have G3P? 3 carbon. And I told you that G3P actually is the precursor to make glucose. One of them, so originally we make 6. Originally we make 6 G3P. And then 6 G3P, why is it become a 5 G3P? Because 1 G3P, you may assume that it goes to make glucose. You may assume that, okay. So the G3P, 5 G3P becomes 3 RuBP. This is the math come from 5 times 3 equal to 3 times 5. Again, the number 5 here is this number 5 here. The 3 here correspond to the 3 carbon in the G3P. Okay? And the 3 here correspond to the 3 here. And the number 5 here correspond to the number of carbon atom in RuBP. RuBP has 5 carbon atom, so that's why it is 5. And of course we need to use ATP to give us the phosphate group. And the phosphate group here is added to G3P, so the RuBP will have a phosphate group in the carbon number 1. Okay, what's so important of the Calvin cycle? Okay, G3P can convert into G3P, glyceraldehyde 3-phosphate. can be converted into other molecule. What are the other molecule? All this. Wow, that's so crazy. Fatty acid and glycerol to make plant oil. Glucose, simple sugars, the major thing. Fructose, when combined with glucose, it forms sucrose. Sucrose is a disaccharide. Remember, glucose and fructose, they are monosaccharide. So glucose plus fructose equal to sucrose. When you have a lot of glucose connect together, it forms starch, or it can form cellulose. Amino acid basically use a glucose, and then add a nitrogen. Nitrogen is coming from the soil, and then you form amino acid. Amino acid is the monomer for protein. I hope you still remember what we learned at the beginning of the semester. It can also make nucleotide. Nucleotide is basically glucose plus nitrogen and phosphate. Plant absorbed from soil, then from nucleotide, which is the monomer for DNA. DNA is a polymer. Basically, G3P makes everything. Everything that we learned from the beginning of the semester. That's how efficient the plant is. Okay, this is another summary. G3P can make glucose, fatty acid, or amino acid. Glucose phosphate can make fructose and sucrose, or excess glucose will be stored as starch in the root, or glucose can also make cellulose as the cell wall for the plant. So basically, G3P is like everything for the plant. That's why it's so important that we learn how to make G3P. Okay, this is the summary of the Calvin cycle. Okay, if we have 6 carbon dioxide, what do we get? We mentioned previously we need 12 NADPH and 18 ATP. Okay, this slide is correct. 18 ATP. So that will generate the carbohydrate C6H12O6. So what happened is that plants, they live in different environments. Some of the environment, they are hot and sunny. They have strong sunlight. It's like living in a desert. In this case, the photosynthesis will become very robust, and it will generate a lot of oxygen inside the leaf. And the oxygen inside the leaf will leave the stomata, but still the production of oxygen through photosynthesis is a lot higher than the oxygen leaving the stomata. So the leaf will have an accumulation of oxygen inside the leaf. When it happens, the excess oxygen will bind to RuBP. Remember, RuBP originally binds to carbon dioxide. But because we have such a high oxygen concentration and the temperature is also very high temperature enhances the RuBP to bind with the oxygen. Then when this happens, we call this a photorespiration. So what happens is that during photorespiration, the overall is that we will make less G3P. So we'll make less G3P. Less G3P. So RuBP can bind with oxygen when the temperature is high temperature, high temp, high oxygen concentration. High oxygen concentration. So it does not bind carbon dioxide, and as I mentioned in the previous slides, it decreases the yield of G3P. Decreasing the yield of G3P is the killing. It's killing.